Okay. So I am listed in the program as presenting a talk called Cancer's IP. That is actually not the talk that I was planning on presenting right now. Instead, I have a talk about this really important patent law issue with respect to biotechnologies that has only kind of recently been considered by a number of scholars in the area. So at the risk of doing something horrifically dangerous, let's take a vote. What do you guys want to hear about? You want to hear about the, you guys want to hear about the biotech, the really important biotech patent piece on which billions of dollars will likely turn, or the cancer stuff? I guess also on which billions of dollars will likely turn. Votes for the biotech piece? Yeah, votes for the cancer piece? All right, very good. We're going to go with the biotech piece, right? That is my empirically sound counting here, right? Okay. One of the problems in biotech patents is that we're really bad at claiming them, right? Biologists study the function of a variety of molecules in biological systems, and if they engineer something remarkable, say CRISPR, they often claim methods and molecules pertaining to that function. But patent law has long cast a gimlet eye on functional claiming. Claims that define their invention by what they do as opposed to what they are. In more precise terms, functional claims prohibit patentees from claiming their invention in functional language, for example, a machine for flying, when structural language will do as well. For example, a helicopter, right? As a consequence, the law surrounding functional claiming for biotechnology inventions is particularly muddled. This is not, and I want to stress this enough, I realize it's 4:20, it's the afternoon, I don't want to put anyone to sleep here. This is not some esoteric question about patent claiming that only the hardest of hardcore examiners at the PTO should care about. It's a question which frequently controls inventions worth billions of dollars. A recent patent dispute between Sanofi and Amgen over an antibody called anti-PCSK9 is but one example of this. This is being decided at the Federal Circuit right now. What I want to suggest here is that the reason that the law is so difficult in this area has more to do with science than it does with law. Unlike other areas of technology, say mechanical engineering, biologists engage in functional claiming because they view the objects of their inquiry in functional terms. Antibodies bind, nucleases cleave, DNA codes. As a consequence, patent law should ideally be able to accommodate some functional claiming in the biotech space to best align patent law's legal layer with its scientific one. This is simple enough. I think it has some enormous payoffs. First, I think it simplifies patent law's antediluvian prohibition on the practice of functional claiming. I also think it complicates recent protests to strengthen patent law's functional claiming as a way of improving patent quality, quote unquote, whatever that means. It illuminates various broader problems in patent law, such as the linguistic difficulty of claim terms, the role of technological complexity in litigation, and the rise of latency doctrines in patent law, and for that matter, elsewhere, right? Or, to put this another way, functional claims have traditionally followed their linguistic form. A superior way of conceiving them is that form should follow function. A brief word about functional claiming and why patent law disfavors it. As with my previous example, the flying machine versus the helicopter, functional claiming seems to permit patentees to claim more than they actually invented. That, by the way, is bad. To claim a universe with the same function as which one thing one engineered, even though that inventor may only have invented a slice of the universe that was actually claimed, right? Again, this is bad, right? This has been recognized as part of the problem, if not one of the principal problems with software patents and with patent trolls. This has been explored at length by Mark Lemley and a number of other scholars in this area. It's also been the subject of the PTO's recent quality initiative. But this is much more difficult in the biotech space, right? This is much more difficult in the biotech space because the things that are often invented are indeed functional rather than structural in nature. So take one case, right? Centocor Orthobiotech versus Abbott Laboratories, for example, right? In this particular case, the court concluded that a patentee's claim to an antibody with a number of functional limitations attached to it, an antibody that binds to a particular epitope with a particular binding affinity, right, was not enough to save the validity of the patent. Although the court did not state it in these terms, it seemed to castigate the patentee for not describing the antibody in chemical structure form rather than its function, right? The particular Centocor case was one about written description, but to the extent that it is requiring patentees in the biotech space to define what their inventions are on a molecular level rather than a function one, this should give us some pause. One of the things I want to propose, this problem has been raised with antibodies relatively frequently, and I guess one of the other things I wanted to propose here is that this is not just for antibodies, but it's actually for a number of other technologies in the biotech space, right? Let's take gene editing as an example. Let's take gene editing systems like CRISPR, let's say, right? Like antibodies, to a certain extent, gene editing systems are defined in part by what they do rather than necessarily what their structural components are. CRISPR-Cas9, for example, is a gene editing system that essentially comprises three parts. A nuclease, this is an enzyme that cuts or nicks DNA. A guide RNA that guides the enzyme to a particular region in the genome, and also, most importantly, that doesn't get kind of written about in the popular scientific literature about CRISPR, something called a protospacer adjacent motif region, or a PAM region. I won't go into the details there. I'm more than happy to chew your ear off about that later, right? If this sounds somewhat vague, right? We've got an enzyme, we've got a motif, we've got a single guide RNA. That's because it is. That's because CRISPR systems are ultimately defined by what they do, whether they, for example, cut or nick DNA, or only a single strand, or both, whether they use one nuclease or another nuclease, whether they respond to one PAM region or another, whether they cut specifically at that particular PAM region or elsewhere in an indiscriminate fashion, as we have seen with a recent development in this technology over the course of the past week, right? Here, a person having ordinary skill in the molecular biology art would define what a CRISPR system is by what the function of it does, right? They would define it by not what its components are on a molecular or a chemical basis, but by what they do synergistically, right? Let's also take DNA, for example, right? Famously, I think biologists tend to think of DNA in functional rather than structural terms. A gene, for example, is a nucleic acid that codes for a specific protein. It is not necessarily thought of by biologists as a chemical compound that has been isolated from the genome, a la myriad genetics, right? Genes with chemical alterations that do not otherwise significantly alter their protein products are nonetheless considered by biologists to be the same gene, right? Not only so, but I think there's relatively widespread recognition among biologists that for any gene, there is no true archetype wild type sequence. That is, whatever we think of as a gene's traditional sequence is simply a statistical vestige of our sequencing of that particular gene across populations. Again, to persons having ordinary skill in the art, genes are ultimately defined by what they code for, not what they as molecules are, right? Currently, in the functional claiming space in patent law, we look to the language of claims in a technological vacuum to determine whether or not they are functional. I think that is normatively wrong. Rather, given that some fields have different views concerning what constitutes function and what constitutes structure, maybe a better way of thinking about this is by asking a person having ordinary skill in the art whether a particular phrasing of a claim puts in their mind the idea that the invention is more structural rather than just functional in nature. In this way, understanding functional claiming as a product of specific technologies, not as a general symptom of patent law's sickness, should cause us to rethink some aspects of functional claiming's recent history, as well as some other greater puzzles in patent law. Here's a couple of these greater puzzles, right? First, this technologist view of functional claiming, I think, complicates the otherwise dire picture of functional claiming that we've painted in the software context. To be sure, right, functional claiming produces some greater ills in the patent system. But as in the biotechnology context, it seems like we need functional claiming to some degree, especially if one of our goals is to align patent law with actual scientific practice, right? Relatedly, this also suggests that reinvigorating patent law's strictures against functional claiming is not going to serve as a panacea for overly zealous litigation that some of its proponents suggest that it will, right? Second, functional claiming has been castigated by courts and scholars by allowing patentees to claim more broadly than their inventions, right? But a technology-specific view of functional claiming suggests that different functional claims across different arts should give rise to different breadths of that technology. Some of those breadths should be permissible, some of them not so. Third, a recent understanding of functional claiming has highlighted that functional claims, writ large, make it too easy for unscrupulous inventors to obtain patents, right? In Mark Lemley's parlance, it's easy to patent problems. It's harder to patent solutions. But I don't think this is really happening in the biotech context. I don't see a bunch of fly-by-night inventors or patent trolls sitting around a coffee table writing antibody patent claims. It's just generally not what's happening in the art, right? Functional claiming aside, this understanding of functional claiming twists and turns some greater puzzles about patent law, right? It underscores the difficulty of simply being able to map words to things, quote unquote. Understanding functional claiming perhaps allows the forgiving linguist in us all to use functional claiming terms where structural ones, for scientific reasons, simply cannot do. It is also yet another example of, I think, biological exceptionalism in patent law. In theory, patent law is technology neutral. Same laws apply whether your invention is in the electrical engineering space or in the biotechnology space. Perhaps this description of the difference in functional claiming here with respect to biotech inventions is just one more brick in the wall of causing us to rethink about just that. Lastly, I think functional claiming shows us the power of doctrinal latency, for doctrines in patent law to essentially lie fallow until they have been recognized by researchers and scholars, right? The prohibition against functional claiming has been codified by statute since 1952. We have had antibody patents since the Wistar patent estate in 1975, but thinking about these two problems as being interconnected until one has not really given us a lot of purchase until just now. So, all right, that's it. I am done right on time, shockingly, and be happy to take each and everybody's questions. Mark. Thanks.
So very interesting. I mean, I think you're absolutely right that the ways in which we use functional claiming differ in the biotech and the software space. I encourage you to distinguish between functional claiming as an effort to describe a category of structures and functional claiming as an effort to expand the scope of ownership, right? And
Sure.
I think
Sure.
you're mixing the concepts a little bit in the
Okay.
talk. So it's not right to say we have a prohibition against functional claiming. We actually have a rule that allows it,
Yeah.
but that says we don't want you to own anything that might perform this function regardless of what it is or how it works. We want you to own only the things that look and work like it. That seems to me the right rule both in biotechnology and in software. The difference in biotech, it seems to me, is really that we have categories of things that we don't understand well enough to describe in structural terms, and so we really are using functional claiming as a descriptor, right? the only thing I know about this antibody is it binds to this epitope with
Right.
this level of specificity. But I think you can both allow that and still distinguish, and there are some cases like the CD20 case
Yeah.
that could say, right, okay, you get to define your antibody that way, but it is this antibody with these particular characteristics that you're trying to define, and when someone comes up with a completely different thing, which also serves the function of that antibody, that's not within the scope of your patent.
Yes, I think you're right, and so we are repeating questions for the sake of the video here. So Mark Lemley at Stanford brings up a really good point that it is not entirely true that we have this writ large prohibition on functional claiming. Is that we have particular limits on when we allow functional claiming or not. Yes, that is absolutely correct. Obviously, I agree with what you say, Mark. For this audience, I didn't know whether it was worth going into the particularities of Section 112, Paragraph 6, and talking about limiting people to what's disclosed in their spec or not, but yes. One of the things that you're right, it would be good to take a look at are differences where functional claiming, even in the biotech context, are used to either, again, broaden the scope of a patent beyond that which was actually invented, something I also agree we should prohibit, and where, it is simply impossible to kind of describe with any additional structural particularity the biotech invention that we have. That's one of the things that I hope to explore in the writing of this paper. So I appreciate making that distinction between those two things. Thank you. Nicholson.
So I wonder how much you think that this is already happening. The reason I wonder about that is the myriad example that you use. So Judge Sweet's opinion, he says, hey, DNA is different than other stuff. It's really about information. We don't care about the structure. Federal Circuit obviously slapped that down and said, no, you're totally wrong. It's all about chemicals. And the Supreme Court seems to go back to the Sweet view and say, hey, what we really care about is the information. So we're not going to kind of dick around with the stuff at the edges. So do you think this is already happening? Might the logic of that spread? Is this a sign of something new, or is that DNA is special and it's only DNA?
Yeah, so I think that there's some, so this is Nicholson Price who had a question about whether or not this is like kind of already happening, especially given the outcome in the lower court opinion in the Myriad Genetics case. Yeah, so I do think that this is happening in some context. I think that there is a problem because there are some instances in which where we have functional claiming problems. The courts don't necessarily identify them as such. And actually, I think the Myriad Genetics opinion is kind of great on that score, right? We say we are not going to allow the patenting of an isolated human isolated human gene, but we are going to allow the patenting of something that codes for the same human gene with respect to the cDNA that's created. And I think it's fair to say that the reason why the court comes out with that decision is because it wrongly considers the difference between those two things to be functional. They code for the same thing. The isolated human gene, the cDNA, they code for the same protein. Biologists would think of them to be the same. To the extent that one wanted to wrap functional claims within the Section 101 inquiry, we should have prohibited both. I think that is maybe a superior way of thinking about it. I am about to be stopped unless we have a burning, super quick question that I can nail. We don't. Oh, wait, Hank, wait, hang on a second.
Who's going to win the CRISPR patent fight?
Seriously? All right, so this is Hank Greeley on who is going to win the CRISPR patent fight.
Other than the patent litigation.
Oh, come on! You know, that was going to be my stock answer. I think with the issuance of Virginia Cheeksness's patent, things are getting really difficult for UC Berkeley. We'll see whether they survive at the patent office post what I think is going to be a loss at the Federal Circuit. There you go.
So I'm Lisa Ouellette. I teach here at Stanford Law School, and I'm going to be talking about work that I've done with Daniel Hemel at Chicago that's part of a larger project about the economics of innovation incentives. I am pleased to say this project is forthcoming in Hank and Glenn's Journal of Law and the Biosciences, and I've had a great experience with them, so highly recommend submitting to them. So the tweet-sized version of our thesis, for those of you tweeting, is that an overlooked benefit of the Bayh-Dole regime is that it allows the US to internalize foreign benefits of its grant spending. Now, there's a lot there to unpack, especially for those of you who are not patent people and haven't thought about Bayh-Dole at all. So I thought I'd kind of start by explaining this debate that we are wading into, with apologies to those who are Bayh-Dole experts in the audience. And for those of you who really want to get into the weeds, this is up on SSRN in draft form, and I can also talk about it more during the Q&A and over pizza and beer at Hank's. So governments use a wide variety of mechanisms to incentivize innovation and to allocate access to knowledge goods. And in practice, these different kinds of innovation incentives tend to operate as complements rather than substitutes. So in this paper, we are focused on mixing of two different kinds of innovation incentives. One is kind of direct spending through grants and national labs funded by US taxpayers, and US taxpayers spend over $100 billion a year on this kind of incentive. And the second innovation incentive is patents, because the recipients of this direct spending can generally patent the results of this research. And most of this patenting is happening in the biosciences. Universities are increasingly being used for the early stages of development of pharmaceuticals and other medical technologies. So the main governing legal regime here is the Bayh-Dole Act of 1980. For national labs, it's actually the Stevenson-Weidler Act, which predates Bayh-Dole but never gets any attention. The basic idea here is that universities and other recipients of federal grants can patent the inventions that result from this funding, with a number of restrictions, including a preference for domestic manufacture of the inventions and a requirement that the patent rents be reinvested in science, research, and education. So this regime has won lots of praise as a huge legal success. It's been called the basis of the US biotech revolution. Proponents point to the large number of products, particularly new drugs, that have emerged through Bayh-Dole and claim that it has produced hundreds of thousands of jobs, saved millions of lives. The Economist, a magazine not usually prone to hyperbole, has called it possibly the most inspired piece of legislation to be enacted in America over the past half century. But there's also been lots of academic criticism of this regime, and it's been increasingly getting bad press, particularly as a result of universities asserting their patents in ways that cause some to refer to them as acting like patent trolls rather than as guardians of the public interest. So here's some headlines of universities winning staggering verdicts from suing lots of tech companies. And universities are also in the news for, we were just talking about this CRISPR patent dispute between Berkeley and MIT's Broad Institute over the genome editing patents. And if you haven't been paying attention to this, you should, particularly Jake Sherkow's work on this. And I think this dispute is interesting because not only does it raise interesting questions as a matter of patent doctrine, but it also is a good time to reflect on why we want anybody to own patents on CRISPR at all, when at both of these institutions the initial research was funded with public money. So why do we let university researchers patent the results of their research? The primary justification that's been offered for this is called commercialization theory. The idea that for some kinds of inventions, having the exclusivity of patent rights is needed to get these inventions commercialized. And this story makes sense when you're talking about a new pharmaceutical where many universities are developing promising drug targets, but the expensive part of the process of getting that drug to market is doing the clinical trials on them. No drug company is going to invest in clinical trials unless they have robust patent protection for them, so they'll exclusively license from the universities. And this theory that you sometimes need patents in order to get something to market, this is the reason that Bayh-Dole was enacted. It's in the statutory text in the legislative history, and it's the main ground on which Bayh-Dole defenders rest their case today. And it's been recognized as the best justification by the many legal and economic scholars who've written about this, including Mark Lemley sitting in the back here. And these prior scholars have argued that the other potential justifications, or justifications of why we have patents in general, just make less sense in the university context, that university scholars don't need incentives to invent, that they're going to disclose things anyway, that universities don't make that much money for, it's not that efficient. So we'll come back to these, but I first want to give one well-known example that illustrates why commercialization theory can't justify the Bayh-Dole Act's present scope, even if we just focus on the Biosciences. And that's, some of you are probably familiar with the Stanford's Cohen-Boyer patents. These are patents on early recombinant DNA technology, basis of a lot of biotech firms. And they were licensed non-exclusively by Stanford to over 400 different firms, raised $255 million in licensing fees for Stanford. But note, they're being licensed non-exclusively, which means that this idea that one company needs to have exclusive rights in order to commercialize something doesn't make sense in that context. Clearly, exclusivity was not required because we have over 400 firms that are using this technology. So note that the concern here is that US taxpayers are then paying twice for this technology. Once when they fund the initial research that leads to this invention, and then again when they have to pay the super competitive price on the patented products. This is an extra cost for both the consumers and the firms that want to use this technology. And since both the raising taxes and having patents creates deadweight loss, we as a society generally only want to do that when we get some benefit that outweighs this cost. And this is not an isolated example. Over 60% of university licenses are non-exclusive, which suggests that exclusivity was not needed to get to commercialize these inventions. So there's two possible responses from Bayh-Dole defenders. One is that perhaps patents aren't justified here, but patents are generally needed for commercialization, and we can't sort them out. I have another paper with Ian Ayres that argues that universities could use a market test to figure out which are the patents like these for which exclusivity is not needed for commercialization. The other possible response is that, well, maybe these patents aren't justified by commercialization, but they're justified by some other combination of potential benefits. So this project focuses on the kind of international considerations, which I think have been largely overlooked in this debate. To the extent that foreign patenting by US universities has attracted attention, it's been from scholars who are concerned about the impact of university patents on access to medicines in developing countries. And that is a real problem. It's something that I've also thought about. But it is not the primary impact of foreign patenting by US universities. We couldn't find any data on the extent to which US universities have sought patents abroad. So we worked with economists at the World Intellectual Property Organization to put this data together. Here's a few entries from a much bigger table that you can get in the manuscript on SSRN. And as you can see, and as is probably no surprise to anyone who spends a minute thinking about the economics of patenting, most university patenting is being done in high-income countries abroad. This is the percent of US patent families that are sought in different jurisdictions. So about a third of patents are also being sought at the European Patent Office. Aside from India, universities rarely patent in lower and middle income countries, almost never in low income countries. So in these low income countries, then for the places where they're actually patenting, the high income countries, the distributive critique about impeding access to medicine doesn't apply there. And note that the kind of paying twice critique also doesn't apply because these consumers in other high-income countries, they also didn't pay for the grants in the first place. Without the patents, they wouldn't pay even once. So from a distributive justice perspective, having patents on these technologies might seem attractive. I suspect that many people would think it's problematic if US consumers bore the entire cost of developing some new drug where consumers who benefit from that drug in Norway, which has a much higher income per capita than the US, paid nothing at all. But I think the even more interesting question is whether such patents also increase efficiency. And here I'm moving beyond what I think we can learn from patent counts to what is really a theory that seems worth more empirical work. So innovation economists such as Suzanne Scotchmer have worried that individual countries will invest too little in grants because there is no form of international coordination on grant spending. So as a first approximation, you might expect a country to invest in direct R&D spending only up to the point where the marginal cost equals its own marginal benefits, which means that it might not account for the benefits that new scientific knowledge has beyond its borders. One way to solve this would be to have a kind of global R&D treaty, a requirement that every country invest a certain amount in grants. We don't have that, but what we do have is an international patent regime. So to the extent the innovations resulting from this grant spending are patentable, countries can internalize some of the benefits resulting from use of those innovations abroad. And note that the Bayh-Dole Act has an explicit preference for keeping profits within the US with its domestic manufacturer provision. So I think it's clear that Bayh-Dole serves this benefit internalization function. What's less clear is whether this in fact increases the amount of direct research funds within the US. In our paper, we discuss kind of two plausible mechanisms by which this might happen: higher federal grant appropriations or greater support from internal university research funds generated from Bayh-Dole patent revenues. On the first point, we don't think that the people in charge of grant appropriations are sitting down and like rationally calculating marginal costs and benefits, perhaps particularly with the current administration. But we think that this hypothesis remains plausible even with a more cynical view of legislative motives. If you think about public choice theory, Congress is more likely to support federal funding when the political benefits are outweighing the costs, and the kind of interest group support for federal research spending will be stronger when well-organized domestic constituencies can profit from these federally funded inventions. The second mechanism is that universities can increase their licensing revenues themselves and have increased direct research funding that way. There are many university tech transfer offices that do not who do not raise more income than their legal fees, but if you consider the system as a whole, the licensing income is rising faster than legal fees. And now, of course, there are lots of caveats to this account, and I've been told to stop, so I'm not going to go into them now. But I want to emphasize we're not claiming that the benefit, this benefit we're talking about here, in fact outweighs the related costs or is sufficient to justify patents that aren't needed for commercialization. And in particular, note that we're only talking here about foreign patents, not domestic ones. Rather, I think this is kind of one piece of a larger puzzle, and that both supporters and critics of Bayh-Dole and those interested in kind of the law and economics of international innovation should be thinking about the role that the Bayh-Dole regime plays in incentivizing knowledge production across borders. So I will stop there and take questions. Yes?
This is Bob Cook Egan from Arizona State. One little wrinkle in this story is you made a little throwaway comment that the U.S. profits are protected by the U.S. manufacturer provisions in Bayh-Dole. And one of the things we're beginning to look into is, in fact, it looks like that's actually absolutely not the case, that U.S. patents are being offshored to foreign subsidiaries. Profits are offshored because the IP rights are leased back for U.S. manufacturer. So that actually the profits end up being an export, and U.S. taxpayers pay for the patents at the front end and pay for the drugs and the products at the back end, but the profits stay offshore. It's a little thing that I don't think Joe Allen or Bayh or ever thought about, but I'm sure you guys are thinking about it, and I'm sure you're aware of that. But how do you even think about something that complex?
Great. So this is a question from Bob Cook Egan from Arizona State, saying that, well, in fact, the U.S. isn't internalizing all these profits because people are offshoring them for a lot of complicated reasons, including the tax implications. And I think that's absolutely right. And so two quick responses. I mean, one, I think one of the audience's take-home messages of this, I think that we need more data, including from the university tech transfer offices, about exactly what's happening in these contexts. That, I mean, when I've talked to university tech transfer offices about like how do you justify these patents that are not non-exclusively licensed, I've gotten a lot of hand waving, and I think that particularly as there's some more negative press about how universities are using their patents, that they kind of need to step up and provide more data on how licensing income and things like that. As a kind of broader theoretical point, I think the way that the people are moving their income around, I mean, it's like, in theory, the US government could have provisions requiring them to, could tax them, and I mean, so it's a policy choice that the government is making that's kind of promoting this. And so would love to talk more about this afterwards, but one minute, so Nicholson.
Quick question. Maybe I can figure it out, but I can't get it, so I'm hoping you'll just tell me. It seems like there's a little bit of a discrepancy between the big narrative of we have this big access problem that poor countries can't get drugs, but on the other hand, you showed a slide that, at least I don't remember correctly, suggests that we don't really care about patents in poor countries because they don't have any money to pay for drugs. So what am I missing?
So most university patents, I mean, are not being sought in poor countries, and so like in the African countries here, we have practically no university patenting going on. There are a few high-profile cases. In the paper, we talk about the Yale D4T case, where it's a patent in South Africa that's created a access problem. This is, I think, one of the points of this paper is that that is not the primary way in which university patents abroad are working, and that people thinking about Bayh-Dole abroad should be focused on what's actually causing the bulk of the revenue transfer. So I'm out of time, but would love to talk more about this over beer.
All right, so I took the suggestion to do early stage talks pretty seriously. This is an early project. I am very much looking for your feedback. In particular, part of me thinks that this is the sort of project that young scholars get really mired in, that seems like I know everything and I want to solve all the problems, and I'm actually totally ignorant and know nothing, and I'm taking on way too much to chew, so I would love your feedback about how to shape this going forward. All right. So with that giant caveat, I want to talk about complicating the mix of biomedical innovation incentives. What do I mean by that? So I think you can imagine that we think about innovation and we think about a problem that somebody's trying to solve. And so a lot of the literature, particularly in the sandbox that I play in, is like, hey, how do patents do this? And then we start branching out. Some are like, how about trade secrecy? And then how about regulatory exclusivity? And then Eve writes a bunch about that, and lots of people write about the patents and the trade secrecy. And then maybe we'll talk about some interactions and some other interactions and more interactions. And so we tend to focus on that. And what I want to do in this project is start to add in more pieces of the picture. So what about when we add in prizes and grants and taxes? Shout out to Lisa for doing a bunch of this, and reimbursement and regulatory approval and publications. And how does this stuff all work together? Obviously, this is way too much. I am not attempting to, well, I would love to develop a grand theory of everything. I am not attempting to do that in this project, but I want to start playing around with how does this stuff work together on the ground a little bit more. And so the way I want to try to at least think about approaching this is to change the scale of this, which is to say, switch from that picture to this picture. Let's look a lot more at one particular innovation landscape and then see how these different incentives are fitting together to drive the behavior of people working in that landscape. And so the question then is, what landscape to pick? And the answer is the one that I thought was the coolest right now. That was actually how this project started. It wasn't like that at all. It was just like, hey, this is really cool. What can I think of to study this cool technology? And so the cool technology that I want to think about is liquid biopsies and circulating tumor cells and liquid biopsies and tests for cancer in particular. All right, so what's the basic problem here? What's this technology trying to solve? For those of you who might not be familiar with it, the three characteristics that I think are key in driving this problem are that cancer is really important to catch early if you can. Perhaps most importantly, it's changing all the time. Tumors aren't static, they're changing. Most importantly, perhaps, they're changing in terms of how they spread and in terms of the fact that drugs stop working because cancers keep changing, and so a drug that's working now might not be working a couple months from now. Also key to this, cancers, particularly non-blood cancers, are really a huge pain to characterize, right? Basically, the problem, I think, could be boiled down to why do we want liquid biopsies? Well, this comparison, right? The top thing, that's the needle that they stick into you when they're trying to get a sample of a solid tumor to use to characterize it. That's not awesome. And often they miss. And that's also not awesome. So this is not fun. You don't want to do this all the time. The second thing, that's just a blood draw. Now, I don't deal well with blood draws, but apparently many people don't have problems with them. And so you can do blood tests a lot more frequently than you can do solid tumor biopsies. So wouldn't it be great if we could use blood tests to evaluate the characteristics of, say, lung cancer or pancreatic cancer or something else? Well, yay, awesome thing, turns out we can. Because we didn't used to think this was the case, but it turns out that solid tumors shed cells that go through the blood, and so if you can find them, then that's great. And then you can do these tests, and you don't have to stick giant needles in people. And so the question is, well, how do you find them? And the challenge, as I heard it described as, it's not like finding a needle in a haystack. It's like finding in a needle made up of haystacks made up entirely of needles. So they're super rare. There's roughly 1 circulating tumor cell per 3 billion blood cells or something like that. It's hard to pick them out. So interesting technology, really important, potentially huge implications for clinical care, really hard, really interdisciplinary, so sounds like fun. Why don't we study it? Why don't I study it? You can go away for now. I have to get tenure first, but I'm going to study it for now. Just kidding. You should all join, and it's going to be very exciting. So this is I'm not going to take you through this, but this is a schematic of a bunch of different ways we can try to do this task. How do we pull these cells out of the blood? And there are a bunch of different things, and part of what struck me is all of the different technologies seem like they're different for, I'm not clear whether they're technologically important reasons or not. So, they'll say, we're going to send it through a microfluidic path, and we'll use little electrophoretic gradients to pull the cells in one direction. We'll use little triangular barriers to do that. And then I'd listen to another group, this is a lot of conference on this thing, and say, we're going to take the cells through a microfluidic path. We're going to use little cross-shaped barriers. And we're going to use a magnetic gradient to pull them. And then I talked to a third group, and they're like, we're going to send these through a microfluidic path. We use little triangular-shaped barriers, or maybe little circular barriers, and we'll use some antibodies, and that's how we're going to do it. It's like, what's going on here? Whoa. So what's going on here? What's actually driving this? Is this really all of these are different cool ways to do it, or are they spreading out because of the way the incentives are set up? And how are the legal structures that we're talking about influencing the way people try to solve this interesting, hard, complicated, important problem? And the answer is, I don't know yet. What I want to do is talk to a bunch of these people and figure it out. So A, if you know these people, please tell me. I'm reaching out to some of them, but I'd like to talk to more of them. B, if you have ways that I can particularly usefully talk to them, I would love your input on that. And so I'm going to talk to you about a few of the thoughts that I've had as I'm thinking about this. I'm not quite done yet, Glenn. I'm not sure if that was a hand or, all right. OK, so a few thoughts. First off, I just started looking at some of the data about publications and grants. You can't really see these, but I'm just excited because I don't usually have data and I miss empiricism. So basically, the thing that I think is most interesting about this is these are logarithmic scales for publications and grants with the keywords circulating tumor cell liquid biopsy, or ctDNA, which is circulating tumor DNA. One thing is these are logarithmic scales that are going way up. So these technologies are getting much more written about in the recent past. This is grants, ditto, going way up. Interesting thing, perhaps not surprising if you've studied, if you've actually been in a science lab, but maybe more interesting if you think about the way grants are supposed to work. The grants lag the publications by about 2 years. So that's weird, right? We normally think about this as, let's give you some money to solve a problem, and then you'll figure it out and you'll write about it. Instead, this is like the pattern that you see if you're in a lab, which is you have to solve the problem and then tell somebody, hey, I should solve the problem. Then they give you money to do the thing that you already did. So that's just kind of fun to see. Yes, they're called grants, but they act like prizes. Right. Query whether or not the system of scientific funding actually results in just widespread forced fraud by lots of scientists. Anyway, so publications and grants getting much more exciting, all sorts of interesting stuff. Also, the first really big kind of corporate move in here happened right around here, just before everything started to shoot up. And also, that's when they got their grants, when they went public, or not when they went public, but when they started becoming much more high profile. Where is this money going? nice, relatively even spread between academia in blue, and then actually a ton of domestic grants going to domestic for-profit industries, especially in the liquid biopsy space, but also for CT DNA, much less for circulating tumor cells, and then a decent amount of intramural work, which is to say like NIH funding stuff within its own walls. I'm not quite sure what to make of this yet, but they're pretty pictures, and so I wanted to share them. So my tentative thought, and I don't know if this is right or wrong, but I'm thinking about it, is that it's useful to think about these incentives for innovation in terms of convergent incentives and divergent incentives. And what I mean by that is some incentives tend to push innovation apart in different directions. Patents do this. They have a novelty and a non-obviousness requirement, so you can't get a patent on something that's been done before. Similarly, publications and grants. How do you get the New England Journal publication? How do you get the giant new grant? You don't get it by doing the thing that the last guy did. You don't get it by doing minor variations. You don't do it by perfecting something. You do it by having path-breaking, groundbreaking breakthroughs. And maybe that's a triangle instead of a cross. I'm not quite sure about that. On the other hand, there are the other types of incentives that I think we don't think about as much, and they're later in the process, at least so far as I've thought. Things like FDA approval, in particular, and insurer reimbursement, which Rachel's written about a decent amount, although she's frowning at me and indicating that I'm wrong, and I look forward to her telling me why. But I think there is something to be said for the idea that FDA gives you an easier time if it's familiar with the technology, right? The whole 510(k) process is about this. Generic drug approval is very much about this. We have ways that it's easier for regulators to deal with things they're familiar with, whether it's formal or informal. Similarly, I think insurance reimbursement is easier if it's not the first technology in the class, if the insurer is already familiar with the type of thing that you're doing, or obviously if FDA has already put its stamp on it. So this is way too busy a slide, but this is my attempt to map out something of a timeline about how this stuff might fit, with the purple things being stuff, oh, I used two different purples. With the lighter purple thing, I like purple, the lighter purple things being things that I think are relatively divergent, and the dark purple things being things I think are convergent, and the green stuff, regulatory exclusivity, taxes, trade secrecy, stuff that I really don't know exactly how it works yet. So I get to do this slide where I had people on my team helping me. I've never done this before. It's very exciting. Sudhana Bharacharya and Jonathan Teets both helped out with this project. And with that, I'm going to stop and ask for lots of help. I'll leave this up there because it's the thing that I didn't talk about. Thanks. I appreciate your thoughts. Rachel.
Well, I love this, and I love all of these projects that try to bring together all these incentives. I think you may be telling a story about devices here that's different from a story about drugs. So Ariel Stern at HBS has some good data about the first product in a class to market versus the second product in a class. And it turns out for drugs, the first product to market gets there faster because they get all these innovation incentives, and then after that they don't get accelerated slower. Whereas in devices, for some reason it's flipped. The FDA has to figure out what is this thing and how do we regulate this new device, and then after that they figured it out, and 510(k) means you get in more quickly. So the story is different in FDA context for devices and drugs, and so I just have a cautionary note about
Right, so to the extent that I'm trying to say, here's this really weird particular technology that's specially about devices, I'm going to generalize about the rest of the world, I think you're right that I should be somewhat cautious about that. Oh, right, so sorry. Rachel Sachs, WashU, said, hey, drugs and devices are totally different because they have different dynamics depending on which one you get first, and I should listen to Ariel Stern at HBS, who has cool data about this. And my answer was before that. Lewis.
Caution to the caution, which is that I assume that the technology you're talking about would actually be Class 3 PMA devices, which aren't subject to 510(k), so they might act a lot more like drugs than like devices.
Yeah, so obviously a decent part of this is going to be working through the specific. Louis Grossman from American University mentions the fact that these are likely to be Class 3 PMA devices, and therefore 510(k) process can't be used until later ones in the process. I agree with you that there is a complicated mix of things going on here. I think part of what's interesting, in additional response to Rachel's question, is the fact that, yes, there are lots of breakthrough incentives for the first people to solve a problem, But those incentives aren't also there for the first people to solve a problem in a particular way. So if you have drugs that are basically doing different variants on the same way to solve a problem, or find incredibly exciting new different ways to solve a problem, you might find that the patent incentives are still there to be quite different. And in fact, we see this with Me Too drugs. But you might also have an easier path in because regulators are familiar, and those, even if those breakthrough incentives aren't there. Yes.
21st century cure is just created a breakthrough devices category.
Right. Yes, Bob.
I don't see, I'm sure it's probably part of your case studies, but are you also looking at angel and VC and influence in the financial models that are driving this?
Bob Cook-Deegan asks whether I am also looking at angel and VCs and additional funding mechanisms, and yes. You know, by which I do in fact mean mostly no, and here's why. To some extent, part of this is an attempt to say here's what's actually happening in the industry, and I recognize that there are a bunch of other things you could try to put in here, right? And to some extent, it's necessarily going to be an incomplete picture because a complete picture is just living in the world. So I think VCs and funding mechanisms come into that. I think things like company culture, like as Jake pointed out when I talked about this at PatCon a couple of weeks ago, you know, some companies just love secrecy. Some companies are like, secrecy is stupid. We're not going to be secret. And yeah, that's a part of this. And that's something that's going to be tough for me to capture. So I'm thinking that it's going to be a useful thing to focus more on the kind of policy levers that we can explicitly drive a little bit more. But it's possible that that's an indefensible choice. And if you think so, I would love to hear that. Yeah, Nate.
So it's
Nathan.
a fascinating chart, and I think it would be interesting. It might make it harder to understand chart-wise, but to understand who is operating at what stage in the timeline. You go from academics, basic researchers, medical centers, you have collaborations, then you have startups, and I think ranging from startups to big pharma companies, medical device firms. So knowing who is operating in which sphere and how it evolves, and maybe, so with the particular technology you're looking at, that seems like you'd be able to get a grasp of that universe. Again, you have the problem of many different types of products if you want to generalize it out to draw some broader principles. But I was looking at something similar in separate research on a different product, but from the perspective of what kind of evidence, what kind of proof or evidence or substantiation do you need at different stages, looking at the very earliest venture capital patent seeking stages and how you prove your product and your technology works along. So I can talk to you about it later, but I think what would be interesting image layer to add to this would be who's doing what at what stage.
So Nathan Cortez points out that my slide is awesome, but it should be more complicated. Which I agree. These arrows should both be broken down into lots of additional things. I am not sure as to what the right breakdown is in the, I'm not sure where exactly on the spectrum I should fall between here's everything and it's super complicated to, I'm a physicist and it's all a sphere. Like, I'm thinking like around here, but I think some people are suggesting I need to be more up here, and I'm also thinking that if I want to generalize more / be fancier, I maybe need to be a little bit more down here, and I don't know that yet. So I'm trying to figure that out. I think that's going to evolve over the course of the project. 2 minutes left. Tanil.
Hey, so one part of the story seems to be specifically for this technology, maybe what's stalling clinical trials is the need to collaborate with physicians who are going to be displaced by this technology. That you can't just have the cells in a vacuum, you have to use them mutation analysis with some confirmation from someone who is not going to have a job if this is approved. So actually, to the one of the first early points you made, my husband's an interventional pulmonologist, and he does this stuff. He does the staging and the diagnosing, but he's collaborating with Parasite or something foundation. They're FDA approved for the mutation analysis, but not the diagnosis. But anyway, you come chat with me afterwards. Complete introduction.
Tanil Brown mentions that it is interesting that you have to collaborate on this type of research with physicians whose job you are intending to take. Point very well taken, and I would love to chat with you after it later. Bruce, I have 1 minute left.
Yeah, so I think one of the things that you showed about the delay in funding is part of that is because, you know, the people, the grants go every 4 years, and actually the NIH for a lot of grants is actually fairly flexible, and I think it's a good thing. So in other words, essentially, if you call your project officer and say, hey, we said we were going to do this, but we want to move, you know, a couple $100,000 in this other direction, you can do that. It's still on the old grant, but it's because of the flexibility that they allow. And then the next grant, when that comes up for renewal, you apply. And so it's different than a contract where you're a contractor and this is the cement, you've got to pour it here, and that's the end of it. It's relatively flexible. And so I would say the amount of the money being spent in the lab is different than the grants awarded to Graph. But that's what I'm saying.
Bruce, whose name I don't know, points out that grants are not contracts. I think quite right, and that maybe I was going overboard when I suggested the system implied that most scientists are committing fraud all the time. I strongly agree with your comment. I did not actually mean that. Please don't, you know, go after me.
Conklin from the Gladstone.
Ah, thank you. Bruce Conklin from the Gladstone. I appreciate that. Thank you.
So thank you to Hank and all of the organizers for bringing us together. As a junior scholar in this field, I just finished my first year at WashU. It's really exciting to all be in the same room, especially, I have to say, with BCD, who has the dubious distinction of knowing me since before I went to law school. So it's really exciting to be here. So my research focuses on questions of innovation law and policy, primarily in the life sciences. And there's a large and still growing body of scholarship about the ways in which different administrative agencies can individually promote health innovation through various incentives they administer. But the thesis of this paper is that we ought to consider the capacities of agencies to act in concert to promote health innovation, and that when we do so, we can achieve new innovation-related goals that we didn't think were possible previously. So in this talk, I'll share with you the general contours of the paper. I'll start by explaining the ways in which 3 individual administrative agencies acting alone can promote health innovation: National Institutes of Health, the Food and Drug Administration, and the Centers for Medicare and Medicaid Services. And I'll then go on to consider the ways in which these agencies do, in fact, coordinate now to accomplish goals more synergistically, and there is certainly some of that. I'll then move on to the biggest part of my argument, which is to envision ways in which these agencies could coordinate, but do not currently do so, to promote innovation into health technologies. And then finally, I'll close by gesturing towards some procedural mechanisms that they might use to collaborate. But I should probably start with a caveat, which is that this is the panel on intellectual property. And ordinarily, yes, these are my people, but this is not a talk about IP or the PTO, and I purposefully put the PTO aside in this paper, and there are substantive reasons for doing so. So within the context of health technologies, these 3 agencies, I think, loom relatively large when compared with the PTO. Second, the one-size-fits-all patent system, at least formally, is a relatively blunt tool for incentivizing innovation, both generally and in health technologies specifically. Third, these agencies have more substantive authority to create and tailor innovation incentives to particular technologies than does the PTO. And then finally, and relatedly, these agencies have a capacity to promote health innovation more broadly than does the PTO, and people can fight me on that in the Q&A, and that's fine. So first, I'm focused on these 3 agencies, which are all key to the process of innovation in health technologies, right? As most of you know, right, we start with the NIH, and their goal is to pay for research. And so every year, they disperse about $30 billion in research funding, and that can drive a lot of innovation. But the research that they fund is of different kinds, and I think this is important. So about half of the funding goes to basic research, right, designed to improve our understanding of particular diseases, much of which is too early stage or risky for private industry to want to take on. And scholars generally view this area of the NIH's focus as a classic public good, right? This is where the government really should be devoting its resources. And then roughly the other half goes to applied or translational research. And here again, the NIH really tries to have a lot of this work complement the role of the private sector in ways I talk about more in detail in the paper, such as by supporting clinical trials on new uses for old drugs. And then finally, the NIH also funds research into something called regulatory science, right, which is basically the study and development of new information, new tools, new methods for use in the regulatory process, particularly the FDA regulatory process. Next, we have the FDA, and most people here are familiar with its role as a drug and device regulator. Many scholars, including some in this room, have written about how the function of the FDA as a regulator is actually one that channels innovation because it results in the production of huge quantities of information about what kinds of drugs are safe and effective for what kinds of uses. But what's less well talked about is that the FDA administers a huge number of policy levers that are specifically designed to encourage companies seeking to develop new drugs and devices. So the FDA has 4 separate programs for encouraging expedited approval for particularly innovative drugs. The FDA awards exclusivity periods, which are a lot like patents for newly approved drugs. And the FDA awards prize-like vouchers for the approval of particularly neglected products. Third and finally, we have CMS, the Centers for Medicare and Medicaid Services. Nicholson mentioned that this is the thing I do write about, and I think not a lot of people in this room do. Because CMS is the health insurer for more than 100 million Americans, in that capacity, it both has a lot of information about Americans' healthcare needs, and it spends a lot of money to purchase technologies to meet those needs. So when CMS makes the choice to purchase particular kinds of technologies, that sends a market signal to companies who then reorient their R&D activity accordingly. So the best empirical evidence for this comes from Medicare Part D, right? When Congress created a prescription drug benefit for many seniors who didn't previously have one, pharmaceutical companies responded by investing more into drug classes with higher consumption among the Medicare population. And this effect has been demonstrated with other coverage requirements as well. But for a while, I think we've thought about this as a fairly linear process, right, where you have a handoff of technology to different agencies along the way. But in fact, there are constant opportunities for feedback and improvement, and we should be asking about the ways in which these agencies can work together to promote health innovation. And the lack of focus on interagency collaboration, I think, is particularly puzzling when you consider that all 3 of these agencies are actually sub-agencies of the Department of Health and Human Services, so there's sort of a built-in infrastructure for them to do that. So when thinking about interagency collaboration, the first thing I wanted to do was construct a taxonomy of how these agencies are in fact collaborating in practice to promote health innovation. And what I did was essentially I combined the taxonomy presented in one of the key articles in the field by Jody Freeman and Jim Rossi with the NIH's own characterization of their collaborative activities. So the NIH is actually required by law to publish a report on this every year, which means they make it really easy for researchers to find all the data on this. It's very functional, and I worry that something is lost when I move from this slide to the next slide, so any thoughts on that would be very helpful. But I've written it a few different ways, and I haven't found the way that seems sort of right yet. And so there's 3 different kinds of collaborative activities that these agencies are involved in. The first is information sharing, in which the agencies bring together representatives to share information between them. And there are more than 50 collaborations devoted to information sharing among these agencies. And this type of collaboration is more prevalent than any other type between the agencies. Although these initiatives all have the same basic function, they differ along a host of dimensions. So not only in terms of the substantive areas they cover, but in terms of what stage are they at within the leadership hierarchy, what's the level of generality, how transparent are they, how were they created, and what's the purpose to which they put the information generated as part of their efforts. On the research front, in some cases, the information generated in the first sort of collaboration is then used by individual agencies in their sponsorship of particular research activities. But in other cases, the collaboration is focused around the research itself. And these initiatives are diverse in their subject areas, but there's much less variety, in the format of the collaborations. And that's in part due to the nature of the research process and then also the agencies involved. So almost all of the research collaborations are between the NIH and the FDA, and they're focused on regulatory science. So they're more similar to each other than they might be if they were between other agencies. And then finally, decision making. So historically, and I'll give the best example of this here because the least developed of the 3 of these areas, Historically, medical device companies have had to navigate two separate regulatory systems. They have to get FDA approval, and then they have to proceed through CMS's coverage determination process to get Medicare reimbursement. But those agencies apply different legal standards at different times to those determinations, and they're concerned with the different aspects of the device. So there's a lot of uncertainty about whether the information that companies generate in the FDA approval process is sufficient to support the coverage determination. So FDA and CMS have a partnership devoted to approving medical devices and making coverage decisions at the same time, but it's not used very often, and it is the least developed of these three areas. I actually thought the program had lapsed, and then they came out with a rule saying, aha, we're finalizing it, and it's only approved one product in 5 years. Great. And I can talk more about this in the Q&A, but there are other things going on here. So one of the things that actually got me into this project was that sometimes the agencies will fight openly about matters of policy, which is sort of strange because if you had a strong HHS head, it's not obvious that you should see that, but it's there, and I'm not sure how much to make of it in the paper. So having considered the ways in which collaboration is happening, I want to consider the ways in which it can and should, but is not happening. And so the goal here is to view FDA, NIH, CMS as agencies with particular core competencies and expertise, and then envision them in complementary situations. And so here I'm theorizing potential collaborations to sort of fill these negative spaces in the existing collaborative landscape. Most of the collaborations we've seen in practice are in this space between the NIH and the FDA, where the agencies are sharing information and then they're engaging in regulatory science research. But in the paper I talk about other potential collaborations they might engage in, and so I'll just give 2 examples in the interest of time. So first, FDA and CMS don't often work together, but there are real opportunities for them to do so in exactly the kind of uncertainty-reducing way I discussed a minute ago. So think back, right, about 7 minutes to when I was describing the accelerated approval mechanisms administered by the FDA in the context of drugs. Those mechanisms are triggered upon a showing that the drug in question is likely to be superior to existing therapies, or it meets an unmet medical need, insert preferred term here. But it's CMS who'd have a lot of information about that, right, as a health insurer. So consultation on questions like that might be beneficial. And then second, I'm very interested in opportunities for collaboration among all 3 agencies, right, at the center of the Venn diagram, because I think that together these 3 agencies have the capacity to serve essentially as a product development partnership in which they set priorities in underserved areas of medicine, conduct research or make grants in those areas, and then shepherd products through the FDA approval process, where the end goal is to approve products which are then marketed at relatively low prices for unmet medical needs. And this isn't aspirational, right? There's actually one thing in the middle right now. It's called the Clinical Isolate Transplantation Consortium. And it does exactly this, right? The idea is that the private sector is relatively unlikely to invest in the development of this sort of cell transplantation as a cure for type 1 diabetes, but it might be hugely socially valuable, and the agencies can collaborate to promote that type of research. In my view, this process should be much more formalized, much more widespread, and the 3 agencies can focus on areas of technology which are neglected by the private sector for various reasons to complement already existing efforts. So I did want to think about why we haven't seen the full range of collaboration and to think about potential procedural mechanisms for encouraging it, to the extent that barriers are practical and not legal. And ultimately, I do think that the barriers here are really practical. There are some laws that restrict the information agencies can share with each other, but they primarily impose procedural restrictions rather than substantive ones. But then there are a lot of standard difficulties, right? Interagency collaboration requires support from senior officials within the agency. There's scarce personnel and financial resources. It may not be worth it from an agency perspective. Agencies may not want to cede decision-making authority or even appear to. But I do think these are barriers that can be overcome, and there's a whole range of procedural mechanisms we can use to encourage the agencies to at least get in the same room and talk about collaboration. So there's actions HHS itself could take, actions that are internal to the executive branch, actions that require the involvement of Congress. And Congress actually has involved itself here, right? They passed a law amazingly enough, right, requiring the NIH to report annually on its collaborations with other agencies within HHS. And so I think there's a lot of opportunity here for this sort of collaboration. Ultimately, I think the choice between these is an empirical question, and I don't have a handle on the practicality of some of these interventions, but I think they can all be used or combined in different kinds of ways. So with that, I'll take questions. Start with Alta.
First, thank you. I think you are very idealistic, and I want to live in your
It's
world.
true.
I really do. I want to just list a few things to take into consideration, and then maybe other people can pick up on it. So a few things. First of all, it's probably valuable to keep in mind that FDA's funding comes from a different committee than everybody else's funding because of historical reasons. It comes out of the Ag Committee. And that makes a big difference in how they all relate to one another in terms of the competition for funding, right? The second is that there is competition among them for authoritativeness. So for example, NIH, which has always viewed itself and is viewed by others as kind of the cool kid on the block, it's the ice cream truck that comes around with all the different flavors, look how great things are. They want to put stuff out as fast as possible. They want to come up with a genetic tests, listing that lists everything that's ever been, imagined in the New England Journal. The FDA, on the other hand, wants only lists of things that FDA has approved. On the other hand, because those tests were coming in as devices and there's no exclusivity, there's no reward, so the companies weren't coming in to get FDA approval. That puts CMS in a funny position because they need to pay for stuff that works. And FDA would rather that they only pay for things that FDA's approved, but FDA hasn't approved anything, so CMS just goes off and does whatever it wants, right? So when you're talking about cooperation, you also have to keep in mind they've got different kinds of goals, which are really very much in conflict with one another. Yes. And I've never known of a secretary of HHS who was capable of actually trying to wrangle these guys. And I will also note for you, when you're talking about unmet need, that the forgotten stepchild of HHS is also the forgotten stepchild of your slide, which is CDC, which actually knows where the unmet needs are and could probably tell these guys where they need to do research, but nobody ever talks to anybody at CDC or pays any attention to them or gives them any money. I could go on and on, but it's a wonderful concept, but I think the political and kind of personnel realities are going to be the biggest obstacle, not the interesting ideas that you can come up with. And so that would be an interesting thing. How can we solve those? Yeah. Right? And good luck.
Alta, let me tell you a story. So Alta loves my youthful optimism, but is concerned that there are structural reasons, such as committee assignments, which I completely agree with, by the way. And I think this is why AHRQ is actually not within the NIH, because it was created by a different committee, which is not, I have had...
And they're never going to give up, the
yes.
committees never want to give up their power.
I agree with this, and I think there's actually a whole paper that needs to be written about this, probably
and
several
NIH
papers.
has a constituency, right?
Yeah.
So patient groups that are their political lobbyists, nobody else has that. So.
And I think this is why the FDA has some of the weird innovation incentives that it has, because that's the only tool the agency has for looking at it. So I completely agree. But I would like to tell you a story of CMS Administrator Andy Slavitt and FDA Commissioner Robert Califf, who when they came into office, they were fighting in public about unique device identifiers and whether they thought it was a good idea or not. And then a year and a half later, they're writing articles together in JAMA about what a great thing it is for FDA and CMS to work together and how we can accomplish so much more when we do that than when they fight. And so I would love to speak to either one of them. I'm aware that I'm on camera right now.
You could probably talk to Rob now.
Oh, yeah. Well, I could probably talk to both of them now. But I think with the right people, with the right agency heads who are committed to this, we could do something. And I don't know that we have the right ones, but I think we can work on it because I am usefully optimistic. Lewis, why am I wrong?
So, no. This is great. Fantastic stuff. There's another barrier that I think you should acknowledge, though, and it's boring but very real. Every single time I've worked on anything concerning interagency cooperation, technological incompatibility of their data systems, it's just unfriggin' believable. I mean, so for example, you could say, oh, why doesn't HHS, why doesn't CMS just share its records with FDA? The answer is FDA is probably using some legacy system from 1991 or something like that.
Oh, it's that modern?
Abacus. No, but just that observation, you should also mention that as a very, very real obstacle to cooperation, even within HHS.
I will. I don't have that in the paper. Thank you. Hank?
So just
Oh.
to-
Oh, it is Hank, so
Okay.
this is the second time Hank has flouted the rules.
just
He gets to do that. Oh, by the way, for the camera, Hank Greeley flouted the rules.
Hank Greeley, conference
aided
organizer.
and abetted by Lawrence. So just to add another thing to Alta's
Helpful
wisdom,
comments.
wisdom of maturity. It's a wonderful idea to have more cost-effective, good, relatively low-cost drugs out there. But I'm not sure you're going to find a lot of pharma companies and biotech companies that are all that excited in that particular result. So I think that may be another political barrier. The industry may not be thrilled about the idea of government agencies getting together to lead to
Yeah.
more efficient drug rule.
I'm being told to stop, and so I will say one sentence, which is that I come from the global health area in which there are different companies with different risk profiles who may be interested in this area. Okay, cool. Thank you, guys.
On the slides up, I want to note that Colleen was the first fellow
Oh, yes.
for Law and the Biosciences. And to have our first fellow having actually finished a relatively lengthy term working in the White House makes me feel really old.
Well, and you know, it was 16 months, but it felt a
Depends on
lot
the White
longer.
House, but this one, yes, proud.
So, well, thanks for that introduction. And the genesis or the kind of motivation for this project came when I was doing this horrible commute, going from California to the White House for this period of time. And what I was really, I think, struck by in this particular area, I'm not, you know, by nature or training a person who's focused on biotechnology, but you know, sort of hearing two things from my two different communities. One is, again, I was sitting in the Office of Science Technology Policy. I was a senior advisor in innovation and intellectual property, and all my colleagues were super excited about precision medicine, about big data, and about sort of personalized medicine. And so you saw all these sort of initiatives come out, the president going on in State of the Union and talking about launching sort of the Precision Medicine Initiative, Cancer Moonshot, et cetera. And then on the other hand, my friends at academia and also sort of in industry, as they were seeing the string of decisions come down from the Supreme Court, most predominantly the Prometheus case, but then also Myriad and then Bilski, Alice, Bilski was earlier, they were saying, oh my God, we think, you know, personalized medicine diagnostics are really important, but the Supreme Court is just basically killing the ability to patent these. And we have this huge set of disconnect, I would say, between these two communities, one which is so excited about getting the science out there and sharing information and really trying to drive the science, and then the legal community saying, wait, but the incentives aren't there. So this project, which I was collaborating with Arti on, was really trying to look at this empirical question of, have these decisions that have made it more difficult to patent diagnostic resulted in a decline in innovation? And we can only observe that looking at patent counts. Trade secrets a lot harder. We can look also at agreements in the SEC, some other sort of indicia. But at this point, we're focusing on those indicia. And then, you know, we sort of looked at this very simple, straightforward, I would say, not as sophisticated as many of the talks that I've heard today, question, but we really just want to see, like, is there a decline? And can we see it in patents? Can we see it also in the narrowing of protection? So just fairly straightforward set of inquiries about whether we see a decline or see a narrowing. And so we tried to test different ideas about what was happening in the market as we were talking to a lot of different I would talk to VCs, I would talk to diagnostic companies. Arti was hearing from different people. I haven't put her name as sort of the co-author on this yet because she hasn't seen all of this, and so I don't want to malign her name with my analysis. But she certainly has been somebody we've been working very closely with on this. So, you know, the question is maybe people are sort of shying away from this area, and so they're moving their money into other sorts of investments. There could be shifting going on where we're moving away from certain, you know, standalone diagnostics, or we're moving to companion diagnostics or something else. You could also say that maybe innovation is still happening, but the protection is narrowing. So downstream, as protection becomes more narrow, there may be less incentive to invest. So how do we look at this? Well, we looked at several metrics in several ways to try to look at the innovative activity. And you know, the key here is to try to find a control and try to find differences with this treatment of the cases. You know, was the trajectory different than it otherwise would have been, which is obviously very hard to tell, but we tried to do our best and just gave it our best shot. So what did we look at to measure innovation? We looked at patent application counts. We also looked at SEC transactions that use the word biomarker. So these are recorded transactions involving material agreements between public companies and a partner that involve biomarkers. What did we look at in terms of the scope of protection? We looked at the actually issuances of patents. So it could be the case that people are filing, but they're not actually getting their patents. We also looked at the scope in terms of the length of the first word. And that's a tricky thing. I'll show you a couple deep dives we did where it wasn't necessarily always correlated with the length. What did we look at in terms of trying to, again, slice between different types of constituents to sort of understand what is happening among the different types of players here. We looked at entity revenue. We looked at the jurisdiction of filing between the EPO and the PTO. We also tried to look at, again, what was happening before and after this 2012 period. The guidelines came out in the middle of 2012, and that's when people really started reacting. So I'm not going to go through the kind of specific CPC codes, but we use those. Those have limitations because they're imprecise, and coding within patent classes is difficult. But these, I think, are fairly good, I would say, best effort areas. And we looked at biomarker innovations, and then we looked at bioinformatics inventions as a control. So we try to create and construct a control, saying other people in biotech, what was happening to them. We also looked, again, as I mentioned before, at trying to look at the EPO and the PTO in comparison. So we'll try to do a head-to-head comparison using a technique called twinning that I and a number of other folks have been using to look at patent outcomes, where you take the same application that was filed in two jurisdictions and you try to trace their outcomes. So if it's the case that the US case law has really made it harder to patent diagnostics, well, you don't have that US case law affecting the European case, but you have the same company, the same patent claim, and so looking at those two pathways can show you a little bit of what that difference is. I'm not going to go through all the assumptions, but there are issues with thinking about what is the lag time that's required to, for an impact like this to take effect and to be internalized. Also, we have lags with respect to the publication of patent applications. It takes 18 months, but there's non-publication requests that can go into effect, so there's some truncation. So there's also some data here that's proprietary, which makes it harder to duplicate. But most of this is open data, and we basically are just trying to acknowledge that these are our approaches and being as transparent as we can. So going into the first question, has innovation declined or shifted based on patent records? What do we find? Well, when we looked at the trend of filings, we found that rather than the sky falling, as you might believe from reading a lot of these patent-specific records and accounts, there's actually still a lot of filing going on. Both applications and grants are going up, and this is based on publication year. So it's, again, there's some lag issues here to keep in mind. Publication year of grants is the actual grant date. So it's a little confusing in terms of thinking about where exactly do you draw that line, but you see an upward trend, and that's something I think that is significant. When you looked at European medical diagnostic applications versus US ones, you also didn't see a relative decline in US diagnostics. You might say, well, US went up, but it didn't go as much up as it did in Europe. Well, you don't really see a big change there in terms of US being the blue line on top, and then the EP applications being the yellow line. On the contrary, the US growth is quite robust. However, when we went ahead and tried to segment by revenue, we did find some validation for the idea that there was, both by revenue and also compared to the control, we did find some validation that even though there was growth, it wasn't as robust as it could have been. So looking at the control group, we saw that there was a big increase in applications from 2011 to 2016, about a 63% increase. And again, that's a much smaller number, so I think it's indicative of something, but I'm not sort of trying to draw a strong conclusion. We're just showing that there are data points where maybe growth would have been even more robust. And if you think about all the funding going towards different initiatives, you can see that there is interest in growing in the sector that is pushing against any limitation on protection. So a 22% increase in the diagnostic applications as opposed to a 63% increase in the control group. In addition, growth, as we found when we try to segment by revenue class, has been uneven. So what was really interesting is first looking at companies were making less than $1 million, and they were doing quite well. And when we dig into the data, we figured out it's because many of them are nonprofits. They make zero revenue, but they're all sort of motivated to keep filing. They're getting a lot of grants. Their growth is healthy. Also, among companies that had over 100 million in revenue, we saw that they were sort of trending upwards. Where there was less consistency with respect to an upward trend is in this sort of startup area of $1 million to 100 million in revenue. And again, this is data that is provided by proprietary sources. I think they pull it together mostly. Of course, if there's public company data, they pull that. I think with private company data, sort of talk to them about their methodologies. There's a couple different ways to estimate that. But these are, those are numbers that are a little bit hard to assess. But still, you see in that band, again, it's a smaller number. You don't see that consistent growth. And so that is something that could be troubling. It could be the people that we're talking to, right? We started talking to some startups that said, yeah, we're really not filing as much. This is sort of their story. But it seems to be outsized in the discussions we're having, but it's not outsized when you look at, it's not really having a huge impact when you look at the entire landscape. So in summary, we haven't found that innovation has strong evidence that innovation has declined after Mayo overall. Relatively or absolutely, but we do see this unevenness with respect to the segments, and we also see that biomarker kind of core diagnostic technologies are growing more slowly than the overall biotechnology, bioprocessing space. What about the scope of protection? Because again, it may be a question of, well, we're still filing, we already had a bunch of investments and infrastructure set up, but as our protection becomes smaller, we're going to just sort of have to change our revenue model. And I will say, and I'll get to that at the end, about the sort of extra, it's really hard to isolate the effect of this case because there's been so many other developments in the space. So when I was doing my interviews, this was really very low down the list of things that they were also worried about. Most of that, the worry was around CMS, reimbursement, and so some other factors that are much more important, I would say, in terms of getting money into the bank. So how do we measure narrowing? We look at claim length. And we argue that if you take a base claim, which is the kind of non-bolded information, then you add a limitation, you add words, that will limit the scope of the patent claim. So all things being equal, a longer claim is more narrow than a shorter claim. However, the problem is that never are two things really equal. So when we dug in and sort of looked at two patent claims actually from the same parent. So these are two twins, one from a US and one EP. This is really interesting to sort of follow this history. You have the EP case quite narrow. I would say the US case is actually narrow in a different way. There's nothing, I don't expect you to read any of that, but it's to say that this technique of trying to look at claim length is also something that's fraught. And so ultimately what we need to do is probably have people read these claims and do some sample work. But if you do look at claim length and sort of look at this question of, well, maybe US patents are being issued, but they're super narrow and they're not worth anything as much. You do see that in both EP and US patents, you are seeing an increase in the kind of word count. And that there is a little bit of a difference, but it's not that significant, right? So you would expect that over time, as land is grabbed, so to speak, in terms of intellectual property, you would have to do narrower and narrower claims to find what's still patentable. And of course, you do see this kind of gradual increase. I don't think we saw anything conclusive here to say that the US patents were getting that much narrower than the EP ones. In general, EP claims tend to be shorter. Again, as I showed you before, that doesn't necessarily correlate to one outcome or another. But anyway, this is, for what it's worth, this is the data that we could pull out. So no clear evidence of narrowing either. Another question, though, that you kind of hear anecdotally is, well, God, it's so hard to get a US patent now, but it's way easier to get a diagnostic patent in Europe compared to the US. And so we wanted to test empirically whether that was the case. And here's where I thought actually doing the case studies going to produce a very interesting insight. And so what we found is when we looked at resolved cases, again, that's a very selected group because you have a lot of pendency and you've got EP procedures lasting longer. So you've got to take that caveat in mind. But if you talk about just resolved cases, then you see that the abandonment rate among EPO cases is actually much higher than among the US cases, right? So if you look at the red line, that's the abandonment rate among EP. The US is sort of, trending. I think the downward trend is partly truncation. But anyway, you see a difference here. And so what's going on here, right? Because the US case is definitely going to be getting these 101 rejections for their first time. That's something that we could see in the data. But somehow that's not ending up in abandonment in the same way in the EP. And so what we found here is, again, no clear evidence here that there was, it was easier to get a patent in Europe. On the contrary, it looked like it's not that easy. But again, because this is very early, we can't really draw strong conclusions. What I think is interesting from my perspective is sort of digging down deeper into these case studies. And so we just looked at 7 resolved cases in the EPO and the USPTO, which all had 101 rejections. So what happened here? When the cases actually do get 101 rejections, does this mean that's the end of that case? So what happened in the PTO? 7 of them got 101 rejections. 5 of those cases were able to overcome the 101 rejection, and you had 2 withdrawals based on 101. So there is a direct impact that's there, but you still were able to get through in 5 of the cases. And there's sort of anecdotal information out there that the PTO is not necessarily kind of enforcing the case law to its fullest. It's trying to carve out some space in their guidelines and that kind of thing. But what happened to the EPO? The EPO had 4 grants, so had fewer grants in the US, but they had 3 withdrawals, and they were based on other topics. One was a 53C withdrawal, which is a subject matter kind of restriction, but the other 2 were based on inventive step and novelty. And here's where I think you get some really interesting finding with respect to thinking about, people are talking now in Congress and elsewhere about let's get rid of 101. It's no longer purposeful. That may be useful because the EPO does not have 101 either. But what they do is much stronger, I would say, non-patent literature consideration, and they really do a very fulsome job of vetting inventive step and novelty. And they're recognized as, they use a panel of judges, of examiners. They use a very thorough process. So you have actually, in effect, fewer patents coming out of the EPO based on this very small sample, but they're getting knocked out on other bases, not on 101, but on other things. So it may be the case on the USPTO side that 101 is sort of presenting some issues, but you can get around them and you can get around prior art more easily. So last thing we looked at was material transactions, SEC agreements with the word biomarker in them. And again, no strong indication that innovation in terms of transactions was going down. That still stayed robust. So again, nothing where I would say it was clearly that things are going up, but I don't think there's any clear evidence of a decline in innovation either. And sort of anecdotally going to these different conferences where there are people in this space and talking to friends of mine that are in diagnostics, they're still trying and trying to adapt to the new landscape, right? So I'll just sort of end with an anecdote or two. So friends of ours that are, one person was working for CareDx, and that is he works to do biopsies after you've had not biopsies, but to try to basically do a blood test after you've had a transplant. My brother's a cardiologist, and so I talked to him about this a little bit. Now, their model was devastated last year when CMS basically cut their reimbursements like by 75% overnight, and that was just completely shocking for the company. They had to go back and sort of lobby and get their rate back up, but it was a really devastating sort of outcome, and this was around the cost effectiveness of diagnostics. So I asked my brother, well, why wouldn't you just adopt this versus doing a biopsy, right? So you have the transplant, heart transplant to a patient, and then why would you want to go and cut them up again? Why don't you just do their blood test? And he said, well, because the regulatory framework is such that you have to go across town to a CLIA certified lab to take this test. You're already compromised having done your surgery, and I want more information. I don't want to know just whether they're rejecting or not. I actually want to know fully what's going on in their bodies. And so obviously these kind of cultural issues, going back to a lot of things we were talking about before, are real obstacles to a diagnostic adoption in general. It's just a really hard business to be in. So I would say that, even though we have this kind of patent story that's going on, it's really a very small piece of the overall one. Thank you. I don't know what time we have. We can take it to the beer if you want.
5 minutes.
Okay, great. Lisa?
This is all super interesting. I'm glad you're looking into this empirically. I have lots of questions. But one is about your choice of control, where so you're looking at updated using the term bioinformatics.
Yeah, I mean, this is Arti's definition. I can show you the classes. I think she just tried to look for stuff where there was still going to be bio. Yeah, the claims do not correlate to disease limitations, but you can sort of look at the definition here. I probably am not presenting it in the best way possible.
Because I think there's many of the same concerns people would have about more
Yeah, with Alice and everything, right.
Arti has a paper on bioinformatics and how they've been getting more scrutiny than more rejected than other kinds of software patents. So.
Yeah, so I think I need to probably be more, I mean, I need to sync with her more clearly on why she chose this. But there was a lot of thought, I think, that went into it, and she felt like it was appropriate. So I will blame that part on her. Mark?
So I think this is really interesting, and the relationship between the law and kind of how people behave, or the lack of relationship within the patent system is really interesting, right? But I think it's also even more interesting to go a step further to the extent you can and try to figure out the relationship between the change in the law and investment money and maybe the ultimate outcome we're interested in, which is kind of
products and stuff.
New diagnostics.
Yeah.
Right? So even where you define, right, which it looks like it's ambiguous, right, that there's change in patentability and change in patenting as a result of the law, that wouldn't be so surprising to me, right? What I'd really like to know is whether the change in patenting in turn affects the actual change in scientific behavior, either on the investment end of the front end or in the outcomes of the back end.
Yeah, no, that's absolutely right.
And the revenue piece you have, although I guess I'm curious how you might disambiguate that from to deal with sort of foreign versus U.S. patenting and the like.
The revenue side with foreign and U.S. patenting?
Well, as I understand what you say is, hey, the revenue growth is less in these industries than it is in the bioinformatics industry.
What I was saying is that, in the control group, the applications are up more than they are in the control.
Too, right?
Yeah, the revenue numbers are with respect to just the straight diagnostic innovation. And what's saying is that, universities are patenting very heavily in diagnostics, so are big companies, and they're still growing. It's those middle companies that are not doing as well. Yeah, Nicholson. Oh, I'm sorry, I didn't repeat the question. Nicholson is asking a question.
Why is everybody so wrong? Like, I think everybody thought, like, this is a disaster. And I heard of these pieces. This is a total disaster.
I think it's because when you talk to the people in the diagnostic industry, there's so much going wrong for them at once that they're just scrambling to stay alive. And this is true of all startups, right? And so I think that, again, if your world is kind of perfect and you have one big change, you're like, oh my God, this is devastating. But when your world is really falling apart in so many different ways, that I don't think this ends up not being what they spend political capital on or what really is keeping them up. They're trying to stay alive. They're innovating and they're doing different things. I think the model of pure diagnostic IP company, like a Myriad company, isn't viable anymore. But these are all adaptive companies. They're just changing their models. And they're really still worried about the reimbursement. That's really a problem for them. Yeah, Jake Sherkow.
Yeah, I have a wonky methodological question. Do you count request for continuing examination as a new application or part of the original application? And there's a good reason why I'm asking that in this context, but I'm just curious as to how you...
Yeah, we're using priority date, so if it's refiled as a continuation, then we're going to recount it.
Cool, okay.
So I don't know if that's a good or bad outcome, but that's sort of what we did.
Getting to Mark's point, I think it would be really, really hard to count the revenue streams using the old codes, but I wonder if the new codes that CMS has put in place might allow you to at least look for last 3 years. Rachel, do you know when they put in the new diagnostic codes for CMS reimbursement?
Well, we're supposed to overhaul the system right now.
But that's going on, but they put the codes in place like 4 or 5 years ago. And you might be able to see, because that would be a much more
granular sort of,
stuff is such a tiny slice
of
what's going on in diagnostics.
And the ACE